I'm sure you're all wondering why we're listening to Vera Lynn singing The White Cliffs of Dover from 1942. Well, actually, the group of organisms we're going to look at today, some of the groups here, contributed to the formation of the White Cliffs of Dover, found on the southern shores of England. So, pretty neat stuff. We'll look at that more in just a little while. We'll return to that old classic hit way back. Well, man, that's getting before our grandparents' time now, isn't it, for some of us? So, guys, let's get into the chapter. We're now in the chapter 28 PowerPoint today. This is online lecture 2D, and we're looking at slides 27 through 42 of the chapter 28 PowerPoint. So we'll start here on slide 27. Basically, our topics today, we're going to be looking at a large survey of many different protist groups. Most of the protist groups that we're looking at today are photosynthetic algae. So remember, protists can be classified just simply by the fact that they're photosynthetic autotrophs. So they're basically algae. We'll look at different algae groups, and then we'll also look at some that are kind of the fungus-like protists towards the end. So should be pretty interesting. Also, I do have a video that we'll look at. We'll look at some bioluminescence exhibited by some of the dinoflagellates, which is one of the groups that we'll look at as well. So let's go ahead and get into slide 27 and really get into our lesson now. Let's look at some organisms you may not be very familiar with. For example, the cryptomonads. Most people are not aware of cryptomonada or cryptomonads. These are unicellular flagellates. Really good picture of one right here. You can see they are one cell in size. That gets them nice and light, and these flagella allow them to swim to different locations. They make up the phytoplankton community. So phyto, because they do perform photosynthesis using light as an energy source, and they float along the top, the surface of the ocean. So they do contain red, blue, green, or brown plastids from secondary endosymbiosis. So they have plastids that are derived from secondary endosymbiosis, which is unlike the red and green algae, which have plastids derived from primary endosymbiosis. So what in the world is secondary endosymbiosis? That would be where you have a cell that has a primary plastid inside, and then that cell with its primary plastid is engulfed by an even larger cell. So when you have a cell that already has a primary plastid, it gets engulfed into a larger cell that is secondary endosymbiosis. And if we had an even bigger cell that engulfed this big cell right here, then you would have tertiary endosymbiosis. But we'll get to that later. These guys are primarily, again, photosynthetic. Again, the importance of them being at the surface, being light, they need to be up there where there's plenty of light. These guys oftentimes are at the bottom of the food chain, as you can imagine, and so they're very important in providing food for small fish and even small invertebrates, small animals, etc. So they do have lots of nutrition, lots of fatty acid-rich food that they store, because they store all this extra energy in the form of these fatty acids that are formed from all that photosynthetic activity. They build up that store, that fuel, then organisms eat them and get lots of nutrition. So very important in that food chain ecosystem there in the ocean. Haptophytes. All right, well, haptophytes, they're also unicellular. They also perform photosynthesis with secondary plastids, and so they're very similar to the cryptomonads. A lot of people believe that they are probably very closely related to the alveolates, straminopiles, and rosaria, which we'll talk about in just a minute. You can see that some haptophytes are known as coccolithophorids. This is a coccolithophorid, guys, right here. You can see it's got a very intricate, beautiful calcium carbonate exoskeleton. These individual discs along the outside of this calcium carbonate shell, if you will, is called a coccolith. So the coccoliths arrange themselves and are glued to each other, forming this shell of the coccolithophorid. So white calcium carbonate discs, coccoliths, eventually develop and form these beautiful, intricate, ornate designs. And then after they die, these haptophytes, these coccolithophorids, settle to the bottom of the ocean and over millions of years can build up to very thick layers. The White Cliffs of Dover, which you see here on slide 27, that song we just listened to, is about this right here, guys. These are the White Cliffs of Dover. You can see the beautiful white, chalky white opened areas. Grass is growing on top of this. Very high pH in this area, which supports a lot of plant life, and does make up a beautiful coastline along southern England's shores. So haptophytes, very interesting, very similar to the cryptomonads, also kind of at the bottom of the food chain. Many organisms eat them. They are just beautiful. Again, with the large accumulations, they do start to look rather impressive. So let's move on now to the next slide, and let's see here where we can find more information on what secondary endosymbiosis looks like. So looking at secondary endosymbiosis, we can see secondary plastids are derived from a photosynthetic eukaryote like a red alga or even green alga. You can see secondary plastids form through the process of secondary endosymbiosis. And I already explained secondary endosymbiosis in the previous slide, where a host cell ingests or retains another type of eukaryotic cell that already has one or more primary plastids, again, something like a red or green algae. So either a red or green algae is engulfed by a host cell. So here's a great picture of what secondary endosymbiosis is like. So here we have the green alga, and here we have a red alga, and they, of course, have primary plastids inside, here and here. They are engulfed by a host cell, and they develop another membrane. And so you wind up with 3 or more membranes on a secondary plastid. You see, guys, every time something is engulfed into a host cell, it gets wrapped by another membrane. Or in some cases, it can even be wrapped by something like an endoplasmic reticulum. So many eukaryotic endosymbionts are often enclosed by a ER, or endoplasmic reticulum. Or it could just simply be the membrane that they got once they got inserted through endocytosis. So now you see what a secondary plastid looks like. 3 or more layers here, as you can see, and you also now know the origin of the secondary plastid, which is secondary endosymbiosis. Again, typically more than 2 envelopes. Oftentimes either a red or green alga has been engulfed to form that secondary plastid. Okay, guys? So there's a little tutorial on that interesting phenomenon here that occurs. Let's go to the next group here, alveolata. Alveolata, all right, this group is highly varied, but the thing that the alveolata have in common, and probably the most distinguishing thing, are these vesicles, if you will, where we have alveoli, or sort of like vesicles, almost like little containers. They all have these sac-like membranous vesicles called alveoli that are present at the cell periphery. So all of the members of supergroup alveolata have these, you have kind of along the cell periphery, you have these little vesicles or sacs all along the outside, along that periphery. We'll look at a picture of that in just a minute. So within the supergroup alveolata, we have the ciliophorans, which includes ciliates. So what do ciliates have? Cilia, right? They swim around with cilia. And do y'all remember, does that allow them to be bigger? Yeah, that's right. Having cilia on the outside can allow your cell to be a little bit bigger. Cilia really work together to move larger cells around. So the ciliates, like paramecium is a good example. A classic ciliate has cilia along the outside, rather large cells, and they reproduce through this thing called conjugation, which is very much like sort of sex. We'll look at that more later on. So we have the ciliates, including paramecium, within this alveolata group. Then we have the apicomplexans. The apicomplexans have an apex at the tip, which they use to invade cells with. So they are medically important parasites. This is kind of a bad group. We think of the apicomplexans as being kind of mean because they cause disease. Plasmodium falciparum is a great example. Plasmodium falciparum, which causes what? Anybody remember? Plasmodium falciparum causes malaria, right? Which kills a lot of people every year. About 2 million people a year are killed because of this guy. 2 million guys. That's a lot of folks dying because of this guy, this Plasmodium falciparum. And we think there are at least 500 million cases. So 500 million people suffer a year because of apicomplexans, mainly in sub-Saharan Africa, places where they don't have mosquito netting. That's where we see the most suffering from this particular organism. Although there are many relief groups that send, missionary groups that send mosquito netting to the folks that live in these areas so they don't get bit by the Anopheles mosquito, which spreads it. Also South America, this can be a big problem. And turns out that Plasmodium falciparum does evolve. It does mutate quite a bit too. So when we get some type of a medicine that treats the malaria, then it doesn't work anymore. So that's a big concern in the future. Anyway, guys, moving past the apicomplexans, we have dinozoa. Dinozoa include the dinoflagellates. Some are photosynthetic, others are interestingly not. So some of them don't act like algae, they're not photoautotrophs. Dinozoa, they have flagella, obviously, so hence the name dinoflagellate, being a big group in here. The dinoflagellate is called red tide. What is red tide? Well, when you have an algae bloom of dinoflagellates, a big population explosion of dinoflagellates, you can actually start producing enough toxin to cause people to get sick and fish to get sick. So dinoflagellates normally produce toxins, but not at levels that would cause illness. But when we have an algae bloom, thanks to the runoff that comes from the land, where we have a lot of fertilizers, detergents, even sewage getting into the water supply, or a river or ocean, we'll see an explosion of these dinoflagellates, and then their toxin, which is normally harmless, becomes an issue because it's so in such high quantities and such high concentrations, resulting in fish die-offs, resulting in shellfish becoming toxic. So if you were to eat oysters or even some of the fish during the red tide, you could get very sick. Causes respiratory problems, neurologic problems, amazing things. I knew a student that was just walking along the beach in Florida during the red tide, and she developed horrible cold-like symptoms, congestion, coughing. Then she even started having issues with her nervous system and nerves, started affecting her ability to sleep, all kinds of things just by walking along the beach during the red tide. Those toxins were absorbed in her body, and she was sensitive to those, making her life pretty miserable. Now, we'll find a lot of dinoflagellates have mutualistic relationships with coral. You see the dinoflagellate, the dinozoan, works with the coral to produce certain compounds the coral can't make. They have a symbiotic relationship. If the dinozoan dies, the coral can die, resulting in what's called bleaching. So the coral are completely dependent on these dinoflagellates with their photosynthetic capabilities. You can see all these guys, again, they're named for their sac-like membranous vesicles, the alveoli along the cell periphery. That's true for all three of these groups. They all have the alveoli vesicle structures along that cell periphery. So a very diverse group. I do want y'all to be able to see something kind of neat about the dinoflagellates here. I'm going to show you just the next couple minutes here. I'm going to show you a video on that. So guys, here are the evolutionary relationships of all the different members here that we've looked at so far. You can see, we've talked about alveolata, including the ciliophorans, apicomplexans, and the dinozoans. They're all, again, similar. We think the apicomplexans and the dinozoans are even more similar to each other, but they're all in this sort of group here. And then we're going to start hitting the Straminophylla and the Rosaria here shortly. So we'll get to those in just a little bit. So there's your phylogenetic tree of these organisms that we're discussing right now. And you can see the critical innovations here that pop up. The alveoli, the sacs along the periphery, which aid in buoyancy and protection. Probably the thing that, you know, caused them or allowed them to survive. Flagellar hairs were very key with Straminophylla and Phyllopseudopodia were very important in leading up to the Rosaria. What I mean by a critical innovation is some type of adaptation that allowed these organisms to become very successful. That's what we call a critical innovation. It's kind of like the equivalent of like a new technology coming out, like the invention of the microchip. And think about what that led to, all these things. Well, these are sort of inventions from an evolutionary standpoint that led to a great deal of diversification and success among these different groups. Okay? So, well, now we've looked at the big picture. Let's go ahead and continue on and look at some actual pictures of the alveoli. So here are the alveoli. You can see they are sacs, you know, vesicular sacs along the cell periphery. Again, allowing for protection. Some of them have been modified heavily to create like an armor, and oftentimes also functions in buoyancy. The plastids, they typically have a placid which allows them to be photosynthetic. Notice all the membrane here in the placid, increasing the surface area for more light absorption. And then some of them have the extrasome. An extrasome is something that is sort of like a projectile. The extrasome is kind of like a little harpoon that shoots out, so that's a defensive thing. So this is definitely a dinoflagellate. Dinoflagellates are known for their extrasomes. Again, these little like harpoon-like structures that shoot out in a defensive manner. Here's a dinoflagellate with the alveoli containing cellulose that appears blue-white. So we have this blue-white sort of structures here containing cellulose which form the periphery of the alveoli. Okay? So there are some really neat members here. You can see some of their neat structures, and we'll get to those in just a little bit. I would like you all to see this video, though. I've got a video that shows bioluminescence of dinoflagellates in the ocean. So, you know, besides causing red tide and kind of making us sick, there are dinoflagellates, again, members of this group Alveolata, that just make things beautiful. So let's look at some bioluminescent dinoflagellates making waves glow at night. Really neat stuff here. This has not been modified, guys. What you're about to see is the real thing. This is really what it looks like when you have a dinoflagellate out in the surf. Absolutely beautiful. So let's see if we can get that to play here.
Perhaps you've seen these videos online. They appear...
Okay, looks like it's not going to let us here. Let me see if we can get this thing to work here.
Perhaps you've seen these videos online. They appear to show glowing blue waves breaking on the shores of some alien...
It's still not quite working here, guys, I realize. Let me see if I can get that to work for us. Here we go. Bioluminescent waves explained. Looks like I waited too long there.
Perhaps you've seen these videos online. They appear to show glowing blue waves breaking on the shores of some alien beach. But this is actually San Diego. To find out exactly what's going on here, we talked to marine biologist Michael Latz at the Scripps Institution of Oceanography at the University of California, San Diego.
Well, this video shows bioluminescence that is coming from a single-celled organism called a dinoflagellate. And in this case, there's really high concentrations of one species, which is called Lingulodynium polyedrum.
According to Latz, this organism is known to form algal blooms commonly called red tides.
These red tides are just a natural part of the ecosystem, but the challenging thing about them is that we don't understand why they occur, and we can't predict when they're going to occur. We know that there's a red tide when we actually see it. You look out on the ocean and you see during the day that the water is discolored. So it looks kind of reddish or brownish in color, but during the night, the organisms in this case are bioluminescent. And this is light that they emit from the cells.
In the wild, bioluminescence acts like a car alarm, going off at even the slightest bump from a predator.
The response by the dinoflagellate is like a reflex. So if it feels a certain amount of mechanical stress, then it's going to respond with light.
So whether it's a predator going in for a midnight snack, or the crashing of waves, a surfer, or a curious kayaker, the results are the same: insanely cool, glowing water. Latz notes that while some red tides can be toxic to people, this one is harmless.
You may not want to go in the water because there's a lot of organic material and you may view that as noxious, but certainly it's really a delightful thing to go swimming at night, and please go with somebody else to be safe, but to swim in it and just observe the bioluminescence for yourself.
For Discovery News, I'm Jorge Rivas, and that's Bioluminescent Waves Explained.
All right, guys, so now you've seen the beautiful footage. Isn't that neat? Some of you may have had that experience. I've always thought it'd be so cool to go fishing during that time, just sport fishing, just to catch them. You could actually see the fish swimming. Imagine, well, I've always thought a really good scene on a Jaws movie would have been Jaws moving through the waters during a dinoflagellate bloom. You could see it glowing as he's approaching. Wow, too bad Steven Spielberg, he could have done a lot better. Or is it Spielberg or who was? Anyway, whatever that producer was that made all the Jaws movies, George Lucas, one of those guys, it would have been a fascinating scene to observe that. Anyway, guys, we'll continue here. Let's continue in here on slide 32 right there. And let's see. Get my highlighter real quick here. Okay, now that's going to pull up. Okay, here we go. Okay, so we are now slide 32, kind of looking at some the dinoflagellates a little bit more. So dinoflagellates, this group we've been just looking at here, again, some of them are heterotrophic, which means they have to eat other things to stay alive. But the other half have photosynthetic plastids, usually from either secondary or even tertiary endosymbiosis. Now, we just talked about secondary, so what would be tertiary endosymbiosis? Well, tertiary endosymbiosis leads to tertiary plastids, and so what happens here is you have like a dinoflagellate ingesting a eukaryotic protist already having a secondary plastid. So here's our secondary plastid with how many membranes, guys? Usually 3 or more, but typically 3. And then that organism is engulfed by an even larger organism, like a dinoflagellate. And so you wind up with maybe even 4 membranes or more when you go through tertiary endosymbiosis. So again, in tertiary endosymbiosis, we're just consuming a cell that already has a secondary plastid inside of it. And so you get lots of layers here. So yeah, that's just about as high as we go. It's kind of like the Russian doll thing. Have you ever seen that? This would be like the big Russian doll, small Russian doll inside that, etc. Again, here's the picture of the sort of the big picture. We'll continue on. And now let's move on to the stromatophiles. So the stromatophiles, very interesting group here. Let's go back there, and now let's go ahead and get that highlighter going here, or pen. So supergroup Stromatophyla. The thing that characterizes this group, so we're moving past the alveolata, we move on to this next group. They have just a wide range of algae in this group, so lots of photosynthetic members, lots of photoautotrophs. We also have protozoans, which are the animal-like protists that are heterotrophic, and even fungus-like protists in this group, organisms that can cause blights to occur, like the destruction of potato crops and water molds, things like that. Now, the thing that they all have in common is they have flagellated cells at least at some point in their life. They're not going to have those flagellated cells forever. But within their flagella, they have straw-like hairs on the surface of their flagella. Very interesting. So you have the flagellum, and then you have these little hairs coming off of the flagella. Anybody know why we would have hairs coming off the flagella? Well, if you have hairs come off your flagella, that grabs onto more water. it makes your swimming much more efficient. So it's really neat. They are, again, heterotrophic or photosynthetic members here, and they typically, this group typically has plastids that were derived from secondary endosymbiosis from a red algae. So during some point in the history of the stromatopylons, they probably went through some type of secondary endosymbiosis. In other words, they consumed an organism that had a primary plastid inside of it already. Okay, so some really neat groups here, very diverse, I would say, extremely diverse group in this particular large supergroup. So let's look at a picture of a typical Stromatopyla member. You can see the flagellum, again, right along here, very long, and then there are the flagellar hairs coming off the flagellum. So we have plastids, again, secondary plastids with, say, at least 3 or more membranes usually, a nucleus, and then food storage molecules. Especially if they're photosynthetic, they usually build up large amounts of food storage in different locations. So that's your typical Stromatopylan. Within this group, we have diatoms. And let's see if we can get that back here. The diatoms, you can see, have very beautiful shells. These shells are very ornate. Lots of details, lots of little pores there. The diatoms actually can filter things quite well. Actually turns out that the diatoms, once they die, their shells can be used as a filter material because water can go through these diatoms with these tiny holes and materials can be filtered out very effectively. That stuff is called diatomaceous earth. Diatomaceous earth can be bought in a pool supply store, so it's very effective. But diatoms are a great example of stromatopylons. At some point, they do have those flagella with those hairs. They actually have shells made of calcium carbonate, but a lot of them have shells made of silica, which is glass. So glass and glass shells and actual glass very similar in chemical composition. Again, that leads to their beauty. So we have both calcium carbonate and glass silica cell walls. Beautiful. They have pores, lines, all kinds of beautiful features. You'll get to see them a lot better. And they also can be used in producing reflective paint, like on the highways. You notice how those lines along the highway and the road you're driving on are just sort of glowing. Those lines are glowing because of the shells, these silica shells that reflect light very, very effectively. Then how about kelp forests? Well, these actually are in this group Stromatopyla as well. Kelp are a totally different type of Stromatopylan, but they do also, like the diatoms, produce a flagella that has the hairs on them. The kelp forest, this is a large brown algae, so kelp are classified as phaeophyceans or brown algae. They are multicellular. They have floats which allow them to stay upright, and they form vast forests which create cover. So this forms very nice cover for all kinds of organisms like fish, shellfish, etc. can hide in these kelp forests, and otters love to go in here and attack, feed on sea urchins, a very common thing. Sea urchins love feeding on the kelp, and then the otter feeds on the urchin. So interesting little ecosystem there, especially off the coast of the Pacific Northwest. So very important in the a marine ecosystem particularly. Okay, now we move on to the next group here, Rosaria. Rosarians, they all have hair-like extensions of the cytoplasm called phyllos pseudopodia. Phyllos pseudopodia. So what does that mean? It's just, remember what a pseudopod is? A pseudopod means false foot. So this is just a very thin false foot that can extend through tiny holes in their shells. So Chlorarachniophyta, Radiolaria, and Foraminifera are your 3 major phyla within this Rosaria group. Again, what do they all have in common? Phyllospseudopodia. So you have sort of like this, you have a shell, and they have little holes along the shell, and then these tiny little Phyllospseudopodia stick out of these holes in all different directions. So they have kind of a spiky appearance. So rosarians typically kind of look like this. And most of them, of course, are photosynthetic plankton. The Chlorarachnophyta are named that because they have spider-shaped cells, hence the arachnio part of the name. These spider-shaped cells possess secondary placids that came from green algae. The radiolarians and foraminiferae are two phyla of ocean plankton. They also produce very beautiful, intricately designed mineral shells. They oftentimes, in case of the foraminiferae especially, are used to infer past climatic conditions. So we can look at the shells of dead foraminiferae, examine their isotopes of oxygen to determine what water temperatures used to be in the past. So we use them in the studying of global warming, these foraminiferae especially. So really neat group, again, mainly shell-covered phytoplankton here in this group. Beautiful pictures here of some of these members. You can see the radiolarian with the phyllopseudopodia shooting out like rays from the sun. Again, these guys are just going to be floating along, performing photosynthesis, oftentimes become food for other organisms. They can die, their shells can build up on the bottom of the ocean too. Foraminiferae here with the phyllopseudopodia shooting out in all directions, just quite beautiful here. And again, these guys used to determine past climatic conditions. Really neat stuff here. Okay, the last group that we're going to look at here in this particular PowerPoint presentation is amoebozoa. Well, actually, we're going to look at the Opisthoconta briefly too, but the last of these, well, I guess you might say the last group is not so much like us. This is kind of a different group here. The amoebozoans, they have all types of amoeba. Amoeba are organisms that simply move around using pseudopodia, false feet. So they are very fungus-like. So when we think of the amoebozoans, you're thinking of things that act a lot like fungi. They have absorptive nutrition, which means they digest food outside their bodies and then absorb that afterwards. They typically have long filamentous bodies made up of long, thin filaments. They are very heterotrophic. They feed on dead things in most cases. And they move very slowly using their amoeboid movement, which involves the pseudopods extending and then pulling the body along. So Dictyostelium discoidum is a slime mold. You may have seen this one. This is a good example. A slime mold is an organism that you'll see in leaf litter. So during the fall, when our leaves start falling, you'll see them also in the spring. But you kind of lift up the leaf litter, kind of dig around, you'll see this orangish yellow material. Kind of looks like vomit. Looks like someone like maybe threw up in the leaves of the forest. But it's actually a slime mold. They're very kind of wet and move very, very slowly, feeding on that decaying leaf matter. Dictyostelium is a great model organism to understand movement, cell communication, and development, where we have a bunch of cells kind of working together as a team to help these organisms function and move. In response to starvation, the slime mold will aggregate into a multicellular slug that develops a stalk structure containing spores. So when things get rough, Dictyostelium will shoot up a stalk structure with a capsule at the top, and then spores come out the top and flow depending on wind direction. Spores pop out and produce new amoeba. So really neat slime molds. I've got a kit of slime molds in lab. Maybe we can grow one, have a little class pet. You can also, of course, find them in nature, very common in this area. So very fungus-like, eating dead things, slowly moving around, using those pseudopods to get around. Okay, really the last group this time, guys, the group that is what we're classified in, supergroup Opisthoconta, includes animals, including us, and fungal kingdoms and related protists. So in this group we have animals, the whole animal kingdom, the whole fungal kingdom, and some protists like choanoflagellates. What in the world do we all have in common? Why are we put in the same group as fungi and certain protists? Well, we all have this thing in common. We all have a single posterior flagellum on our swimming cells. You're probably thinking, what? We have a single posterior flagellum on our swimming cells? Yes, on human sperm, we have a single posterior flagellum. And so we're thrown in this group. And there are even DNA similarities. And we do find genes and proteins that are only in this group, not found in other groups. So there is some evidence that maybe there is some relationship here, and there is relatedness. So we have Cryptomonada, kingdom Animalia, fungi, all in this large supergroup, even Nuclearia. So supergroup Opus thecodonta. Let's now look a little bit more at this group here, see if we can get a good picture of these guys. Let's back up. Yep, there we go. Let's look at the choanoflagellates. Since we're talking about protists here, let's focus on the choanoflagellate protists. Choanoflagellate protists, of course, have a single posterior flagellum, as you can see here and in green here. Now, around that flagellum is a collar. So we have this collar that goes along here and kind of hooks onto the top there. So the flagellum twirls and spins, bends, and straightens. This creates a water current. The water current sucks all kinds of materials through the collar. And so the collar acts as a filter. So think of this collar as just a filter. It's going to filter out food from the aquatic environment. So choanoflagellates are typically sessile, meaning they don't swim around. They stick to a surface like a rock. They often form colonies, and they wiggle their flagella, creating water currents, and simply filter feed. They all have a distinctive collar, and that's why they're called choanoflagellates. Choana refers to the collar structure. They are the most modern protists. They're the most related to animals. So these are the modern protists most related to animals. Most people think that a choanoflagellate-like organism existed in the past and eventually evolved into fungi and animals. So some people would say we had an ancestor that looked a lot like this. Again, from that evolutionary perspective. So choanoflagellates are thought to be, or something like them is thought to be the ancestor of all fungi and animals. Pretty striking. But again, that single posterior flagellum does provide some evidence that maybe that could very well be true. Guys, this is a nice summary here on slide, this table 28.1. Just a nice summary of all of the major supergroups. Again, we looked at, in this particular lecture 2D, online lecture 2D, we looked at Alveolata, Stromatophylla, Rosaria, Amoebozoa, and Opus thecodonta. And we will, in class, talk about Excavata and the land and plant algae organisms here, these green, red algae, etc. So we covered quite a bit here, this group here in this table. We looked at most of the supergroups, about 5 out of the 7 of them. So we covered a lot. Well, guys, we're just about done here. We'll go back to Vera here, let her kind of close our lecture here. And so let's go ahead and listen to the rest, or at least portions of the White Cliffs of Dover from way back, way back in 1942.
Tomorrow, when the world is free, the shepherd will tend his sheep, the valley will bloom again, and Jimmy will go to sleep in his own little room again. There'll be bluebirds over the white cliffs of Dover. Tomorrow, just you wait and see.